Hello, today I'm just going to talk about the potential solution of how to stop improper use of abdominal x-rays for abdominal pain in any settings. So when I was a student on placement, a radiographer who was supervising me at the time said the following to me, and that got me thinking if AXLs are actually useful. So I conducted a literature review and found that there is a guideline for AXL requests, but only 32% of A&E physicians were actually aware that there is a guidelines, and it is very important to only request an AXL for pathologies that are listed on the guidelines, because only 9.4% of unjustified AXL will show a significant finding, while 76.7% of justified AXL will show significant findings. So an algorithm made with BMJ best practice guidelines will suggest if a patient warrants an AXR or not, and the doctors can decide whether they would like to go ahead.
Hi, my research focuses on positron emission tomography data, PET for short. Whereas PET provides the functional information, it can be aided with the anatomical information provided by CT for defining tumor volume. The traditional way of capturing images and clinical diagnosis per image is hand segmentation by human experts, which is very time and money expensive, in the order of 2,000 images per patient. That is the reason why computational algorithms for tumor segmentation come into play. The basic rationale behind our approach is kernel smoothing method. You can treat a 2D image as a matrix and the intensity values as our observations. And we put weight to each pixel according to the closeness, therefore incorporating spatial information. Here is the segmentation results for tumor region on the right-hand side, and the percent volume contour is also provided below, which gives us the percentage of the tumor volume. If you want to know more, don't forget to visit the number 2 booth on the day of ASM 2021. Thank you.
My name is Agnes Lene and I'm from the University of Edinburgh, and this year I am presenting a poster on 6 differences in TSPO expression in the murine brain that were identified using LW223 PET. Several recent studies have reported that in a healthy brain, TSPO expression differs between males and females. We aim to explore whether LW223, a novel TSPO radio tracer, was capable of detecting these differences in vivo. In order to do so, we injected healthy male and female mice with LW223 and performed dynamic PET scans using micro-PET CT system. As seen from the parametric image on the right-hand side, male brain had significantly higher LW223 uptake when compared to females, suggesting higher TSPO expression. Importantly, we were able to measure regional differences, which was not performed in the previous report. If you would like to find out more about the study, please visit the poster number 3.
Hello everyone, my name is Philip and welcome to my lightning talk about my poster about brain networks and challenges of high correlations in these networks and how to overcome them. Brain networks are typically represented by correlation networks such as the simulated one you can see here on the top left. These correlation networks come with the challenge of representing not only direct but also indirect links in the network. These indirect links can lead to drawing false conclusions, so we want to filter them out. To better understand this, have a look at the box at the bottom left. Assume we have 3 nodes, A, B, and C, and A is directly coupled to node B and C. If we were to measure this network by correlation, we would find a correlation between node B and C, which is then an indirect link because it is just arising because B and C are linked to node A. Existing methods to filter out these indirect links fail in the presence of high correlations, and this is typically the case in brain networks. We provide here a new method which is able to deal with this case. And if you want to find out more about this, come to my poster number P4.
Hello everyone, my name is Shay Roddy. I'm one of the radiology registrars in Dundee. Thanks very much for checking out this lightning pitch for my poster presentation, which, along with the colorectal surgical colleagues, we aim to see if there was a link between preoperative calcification on CT in those undergoing potential of curative colorectal surgery. We took 50 patients in those who were undergoing colorectal surgery, and we looked at their CT scans and graded their coronary artery calcification and took into account some patient risk factors and graded it as mild, moderate or severe. Surprisingly, there was actually quite a few patients who had severe coronary artery calcification in those who had no risk factors. And this is important going forward, as it could potentially tease out those patients who may not be suitable for surgery. Check out my results at the poster on the day. Thanks very much.
Hi everyone, I'm Sydney Williams, and today I'll be comparing two SAR management methods for parallel transmit MRI at 7T. 7 Tesla MRI is notable for increased SNR yielding higher spatial resolution, yet comes at the cost of poor RF field uniformity shown on the left. Parallel transmit, or PTX, combats this by using a specialized coil driving independent RF channels to generate a uniform excitation as seen in this gift. Due to the summation of RF pulses, the superimposed electromagnetic fields could potentially cause unintentional heating, also known as the specific absorption rate SAR. In this poster, we investigate the SAR management of two 7T PTX coils, a commercial one and a self-built one by Shajan Gunamani here at the University of Glasgow. The commercial coil manages SAR with hard power limits on each channel. The self-built coil manages SAR using the electromagnetic field simulations done by my colleague Paul Michelini. Both PTX coils produce excellent image quality. The notable difference is the much lower SAR values with the self-built coil. We furthermore assert that these lower SAR values are more accurate since they are derived from the exact coil model. Thanks, and learn more at my poster P9.
Hello, I'm Liam, and the title of my poster is Drug Risk Assessment and Repurposing Using Biomimetic Chromatography and Body-on-Chip Technology. So, the drug discovery process is currently quite lengthy and expensive with really high rates of attrition, and this highlighted area here in particular has quite a large room for improvement. So our aim is to address some of these preclinical shortcomings during the lead selection and optimization stages by combining the body-on-chip derived pharmacokinetic and pharmacodynamic data with data obtained from biomimetic chromatography in order to produce a novel in silico model for drug risk assessment, and then validate the predictors of blood-brain barrier penetration and drug distribution using pre-existing in vivo human PET data. So I'll be around in the first half an hour of today's lunch break to answer any questions you may have on my poster.
Hello, my name is Esma. This study is part of a larger project on the application of machine learning techniques for the prediction of dementia using routinely acquired NHS data. In this study, we have applied a machine learning pipeline on a small clinical dataset. The implementation of the whole pipeline was achieved inside a safe haven environment provided by the Health Informatics Center. You can visit their exhibition booth at this conference. Initial results show high classification accuracy when applying the chosen pipeline, even on a small heterogeneous dataset. Future work will include the application of the pipeline in the national safe haven with a larger population and incorporating other types of data such as genomic and clinical. For more details, please visit our poster session. Thank you.